So what I'm going to talk about tonight, or try to talk a little bit about tonight, is what scientists really mean when they say mutation. So I presume that a lot of you are here tonight because you want to know what happens when this meets this under some shady circumstances, which I can't speak to because I'm not really a physicist. So if you haven't seen the film and you don't want to know, look away now. This, and with a further phenotypic progression to this. So I hope you won't be too disappointed if I tell you that this is not really how genetics and mutation work. So what I would like to do tonight is try to give you an idea of what it is that scientists really mean when they talk about genetics and mutation. So this is DNA, deoxyribonucleic acid. This is the genetic material of all life on Earth. And for being such an important molecule, it's a relatively simple molecule made up of only carbon and nitrogen, hydrogen, oxygen, and phosphate. And together these elements combine to make what we call nucleotides, and these come in 4 varieties, A, T, C, and G. Now, it's the specific interactions between these nucleotides, G's pairing up with C's and T's pairing up with A's, that hold the 2 strands of this molecule together. And this creates not only a very beautiful molecule, the double helix, but, importantly, this provides a built-in mechanism for your genetic material to accurately and faithfully replicate itself so that it can be passed on to all future cells. Each strand can serve as a template to guide the replication of the correct cognate strand. So DNA is your genetic material. It has the ability to replicate itself. What is the connection then really between DNA and genes? So what are we really talking about when we say genes? What is a gene? Does anybody want to take a stab at what is a gene? Some of you know. Well, I'll give you my definition of a gene for tonight in a second, but something I want you to think about, and I may remember to come back to this later, and I may not, but you can think about it anyway. Only about 1.5% of your 3.2 billion nucleotides of DNA could be called genes. So there's a lot more to your DNA than genes. All right, but for tonight, we're going to define a gene as a discrete region of DNA that encodes the sequence of a specific protein. So we're going to say that genes encode proteins. So to take this a little bit further then, if you think about your DNA as instead of the instruction manual for the function and organization of an entire living organism, for instead the function and organization of your home's plumbing system, then you could perhaps think of these as the proteins that are able to carry out that task. These might be mutant proteins, I'm not sure. Now, each cell in your body contains thousands and thousands of different proteins, and it is the specific kinds of proteins that each cell is making at any given time that tells that kind of cell what kind of cell it is. So if you were a plumbing cell, a cell of your intestines, you're making proteins, you need to be that kind of cell. If you're a carpenter cell, a bone or a cartilage cell, you're making proteins that enable you to be that kind of cell. Or if you're a general contractor cell, a brain cell that tells all the other cells what to do, you're again making proteins specific to that cell type. So this is the connection between genes and proteins, but in order for us to get from genes to proteins, there's one more tiny step that I need to tell you about, which is we need an intermediate to carry the message from the book to executing the project. And that message is RNA. So in order for a gene to be successfully, its information to be successfully made into a protein, discrete regions of DNA that are genes are first transcribed in a process called transcription into short molecules of RNA. And it is the RNA that carries the message of the gene in order that a protein can then be synthesized. So why do we need this intermediate step? This seems confusing to people. Why can't you just have proteins in DNA? Well, one way to think about it might be, if you have a textbook for a class, and you need certain pages from this textbook in order to write a paper for your class, the only copy of that textbook is on reserve in the library. Oh no. But you always do your best work in your dorm room with your computer. What will you do? Well, you can go to the library, you can photocopy the pages that you need, and you can take them with you to your dorm room to write your paper. So, you can think of it this way. DNA is isolated in the nucleus of every cell in your body to keep its genetic information safe and sound. And the relevant information that a given cell needs at a given time is transcribed into RNA molecules, which can then go out into another cellular compartment called the cytoplasm, where protein synthesis occurs. So the final step in this process, once we have our genes being transcribed into RNA molecules, is the synthesis of proteins. And this process is called translation because we're actually undergoing a switch here in the molecular language that we're using. DNA and RNA are both made of nucleic acids or nucleotides, whereas proteins are made of something else. Can anyone tell me?
Amino acids.
Amino acids, yes. So we have to switch languages from nucleic acids to amino acids, and that's why this is translation. This is the most amazing slide, even though it doesn't look like it. We'll come back to it later. This is your genetic code. It's also the genetic code for everything on Earth that has genetics. And what this enables, this isn't what your cell's looking at. In order to make a protein, this is how we interpret the genetic code. We can look at it. This enables us to look at a short sequence of RNA. I should also tell you that RNA has U's instead of T's, so that's why you see U's here and no T's. You can use this genetic code to interpret a short sequence of RNA and determine what sequence of amino acids would result in a protein that was synthesized using that RNA's information. So for example, for this first 3 letters of RNA, A, U, G, that would give us a methionine. And if you're in my cell biology class, you have to do this on your exam next week, so pay attention. So we can further proceed through this. C, G, A would be an arginine, and et cetera, et cetera, et cetera. So we have a complete chain of amino acids for a given protein. Now there are also some 3-letter codes of RNA that say when you've reached the end of a gene and therefore reached the end of a protein sequence, and those are designated as stop codons. Step. Okay. So something that's really important that I think about a lot as part of what I think about in my research, but I don't really have time to talk to you about tonight. Just going to give you a few examples, is that once we've made a protein, that's not the end of the line. Once a protein is synthesized, something very important has to happen. Newly made proteins are all synthesized as a linear sequence of amino acids. But in order for those proteins to then go on to perform their function in the cell, they have to fold up into a complex and beautiful 3-dimensional structure in order to do so. This is the 3-dimensional structure for a protein called green fluorescent protein, which we'll come back to in a little while. So scientists have known for a very, very long time that it is the sequence of these amino acids that determines the structure of a protein, and it is the structure of a protein that determines its function. So you can envision that a change at the level of the DNA in a nucleotide could result through transcription and translation to a change in the amino acid sequence of a protein. And this would be a mutation. This mutation will interfere with the ability of this chain of amino acids to find its correct structure and therefore to do its correct function. So instead of having a correctly folded protein, you may instead end up with alternate conformations that have adopted untenable structures. And just to put this into a little bit more cartoony context for you, because this is not cartoony enough. For example, if in order for a protein or an enzyme to perform its cellular function, it must interact with another protein, and both of these proteins are correctly folded, they can form the correct complex to do the function that it needs to do. However, if we have a mutation and protein A now adopts a mutant conformation, can no longer interact with protein B, no protein complex, no activity. This is an example of something called a loss of function mutation. This is important in many human disease states. Alternatively, the converse could happen, where mutant protein A adopts an alternate conformation that enables it to now interact with some other molecule in the cell that changes its function. This could be bad. And this is an example of a gain of function mutation, which is another important contributing component to many human disease states. So this is one way that scientists think about mutation. Now, I told you a while ago that only 1.5% of your genome actually is genes. What's the rest of it doing? A lot of stuff. Some stuff we don't know. But some stuff is regulatory functions. In order for a brain cell to be a brain cell, and a kidney cell to be a kidney cell, and a bone cell to be a bone cell, they each have to be making only the proteins they need. Which means you need to be transcribing only the genes that you need to make those proteins. You don't go to the library and photocopy the entire book that's on reserve and take it back to your room. That would be a colossal waste of resources. Same thing for your cells.